Assalamu alaikum guys, welcome back. Okay, so in this lecture we are going to talk about the in detail in about the structure of hemoglobin, and in the previous lecture we talked about the structure of myoglobin in detail, and this is another heme protein called the hemoglobin. In fact, this is the most abundant source of heme in the body. It is found inside the erythrocytes, the red blood cells, most abundant source of heme in the human body. Okay, because erythrocytes contain each erythrocyte contain hemoglobin, and erythrocytes are the largest number of cells, single cell type in the body. Around 25 trillion RBCs are there in the body. All right, so each RBC contains hemoglobin, and the main function of hemoglobin is oxygen transport. And this is also a heme protein, hemoglobin. Okay, and we've discussed the structure of heme as well in the previous lectures. I've given the link to that in the description. Do check that out if you haven't. And the type of hemoglobin which is found abundantly in adults is hemoglobin A. Right, adult humans contain hemoglobin A as the most abundant source of hemoglobin. And this hemoglobin A contains four polypeptide chains, two alpha and two beta chains, or alpha and beta subunits, and they are held together by non-covalent interactions, and these interactions are predominantly hydrophobic interactions and ionic bridges or ionic bonds between the polar amino acids, and hydrophobic interactions between the non-polar amino acids. Each chain has a hydrophobic heme binding pocket that stretches the alpha helices similar to those found in myoglobin. Okay, in the previous lecture we studied the structure of myoglobin, and myoglobin also contain hydrophobic heme binding pockets. Okay, this was the structure that we discussed in the previous lecture, and it contained hydrophobic heme binding pockets. Why do we call this hydrophobic heme binding pockets? Because all the amino acids which are in these two helices, they are hydrophobic, with an exception of these two amino acids. With an exception of the two histidine molecules, the rest of the amino acids which have their R groups facing towards this heme group, they are hydrophobic, they are non-polar. And inside of these heme groups, we contain an iron, a heme molecule, uh, basically an iron and a protoporphyrin ring, which is bonded to an oxygen, and an E7 distal histidine and an F8 proximal histidine. F8 proximal histidine is directly covalently bonded to the iron, ferrous ion, whereas the E7 distal histidine, it is bonded to the oxygen, and its main role is stabilization of the bonding of oxygen to the ferrous ion. Similar structure is also found in a hemoglobin molecule, but the difference is that myoglobin contains only one chain, so this means that only one heme group is present, whereas in hemoglobin we contain four chains are present, two alpha and two beta chains are present, so four heme groups are present as well, each chain containing its own specific heme group. Okay, and heme binding pockets are there. It is more complex than myoglobin, and yeah, and one more function of hemoglobin is that to some extent it transports carbon dioxide and protons as well. Protons, basically the H positive ions, these are also protons, and carbon dioxide, they can also bind to hemoglobin and can be transported from tissues to lungs, and can transport four molecules. And yeah, hemoglobin can transport four molecules of oxygen, okay, four O2s, and whereas myoglobin transports only one, actually doesn't transport, but rather it stores. Its primary function is to store oxygen molecules. Okay, so each since myoglobin contains only one polypeptide, so it can store one oxygen molecule per molecule of myoglobin. Okay, and it transports, myoglobin provides the oxygen when it is needed by the muscles. Okay, but otherwise it stores it and does not allow the oxygen to leave it, whereas hemoglobin, it can transport four molecules of oxygen, each of the chain transporting one oxygen molecule. Moving on to the structure of hemoglobin, this is how it looks like. Okay, it contains an alpha subunit, alpha 1, another alpha subunit, alpha 2, beta, and two beta subunits, two alpha and two beta subunits, beta 1, beta 2, alpha 1, alpha 2. All right, the alpha subunits are identical to each other, and the two beta subunits are identical to one another. All right, in between the alpha 1 and beta 1, there are strong, extensive hydrophobic interactions. Extensive hydrophobic interactions are present. Hydrophobic interactions are present. Interactions. Whereas in between uh, the dimers themselves, in between, okay, so this goes for alpha 2 and beta 2 as well. Alpha 1 and beta 1, this entire structure collectively is referred to as a dimer of hemoglobin. It's called a dimer of hemoglobin because it contains two polypeptides. Di means two polypeptides, and this is one entire dimer of hemoglobin. And this is the second dimer of hemoglobin. Okay, so it contains hemoglobin contains two dimers, alpha 1 and beta 1, and alpha 2 and beta 2. The bonds within the dimers in between this specific polypeptide chains, alpha and beta chains within the dimer, they are very extensive and strong hydrophobic interactions are present, whereas the bonds in between the dimers themselves, these bonds, these are relatively weaker than the hydrophobic interactions, and these are ionic bridges or ionic bonds, ionic and hydrophobic interactions, but these are less extensive. These are mainly ionic bonds and they are less extensive. And the entire hemoglobin structure contains what non-covalent interactions. Okay, so ionic bonds and hydrophobic interactions are present. And these are referred to as dimers because when oxygen molecule binds to them, to any one of the heme prostatic groups. These rings over here, these are representing the heme groups. Okay, these rings, there are four rings in present inside the entire hemoglobin molecule. These rings are representing the prostatic groups, and when oxygen binds to any one of the prostatic group, it interacts with the other prostatic groups and induces a change in the entire structure of the hemoglobin. Okay, it actually cannot directly interact with them, but rather it induces a change in the polypeptide chain around it, and this polypeptide interacts with the adjacent polypeptides, and due to which the polypeptides interact with the heme group then, and so this is how the heme groups interact with one another, but through an indirect fashion by the intervention of the polypeptides. And so this entire dimer, it rotates relative to the other dimer when it gets oxygenated, and that conformation after it is oxygenated is known as the R conformation of the hemoglobin, meaning the more relaxed conformation, and that R conformation can easily accept subsequent oxygens into the other heme groups. Okay, and other than that, if oxygen is removed from the hemoglobin molecule, then the strong bonding occurs between the dimers themselves. They become tightly compact, and the deoxygenated structure of hemoglobin molecule is the T conformation of the hemoglobin molecule. T means tense conformation. Tense means it is tightly bound to one another, and R means relaxed conformation, meaning the polypeptide chains are relaxed and they are not as tightly bound to one another as they are in the tense conformation. So it's difficult for the oxygen molecule to attach to the hemoglobin in the T conformation, but it is much easier for the oxygen molecule to attach to the R conformation. So we can say that the R conformation has a much higher affinity for oxygen as compared to the T conformation of the hemoglobin. Okay, and this change in conformation of the hemoglobin is known as cooperative behavior of hemoglobin, and we are going to discuss the cooperative behavior in the upcoming lecture, okay, in detail. And the entire dimers, they are identical to one another. Okay, so I can say that alpha 1 and beta 1, this dimer, and alpha 2 and beta 2 dimers, they are identical to one another because the alpha subunit, alpha 1 is identical to alpha 2 and beta 1 is identical to beta 2. So therefore, since the individual subunits are identical, so the two dimers are also identical to one another. Let's move on to the structure. Let's talk about the structure of hemoglobin in a bit more detail now. Okay, it is I've listed down all the points that I discussed in the previous slide. Okay, so tetrameric hemoglobin is composed of two dimers. Okay, alpha 1 beta 1 and alpha 2 beta 2 are the two dimers which are present in the tetrameric. The two polypeptide chains within the dimer are held tightly primarily by the hydrophobic interactions, and We've talked about that as well. The two dimers are held together by polar bonds, which are weaker than the hydrophobic interactions within the dimer. Well, this doesn't mean that polar bonds are always weaker than the hydrophobic interactions. Okay, this is the case because the hydrophobic interactions in between the within the dimer are more extensive. There are greater number of amino acids which are present here to bind with one another. So that's why they give them us a stronger strength, a greater strength, or makes them more stronger there. Whereas in between the adjacent dimers, there are lesser number of polar amino acids. So that's why those polar amino acids have formed less extensive polar ionic bonding as compared to the more extensive hydrophobic interactions. Okay, so that's why the hydrophobic interactions within the dimer are stronger than the polar bonds between the dimers. Okay, so the I could say that the intramolecular, intradimeric means within the dimer, in between the two polypeptide chains, the intradimeric interactions, the hydrophobic interactions are stronger than the interdimeric. Interdimeric means between the dimers. Interdimeric. Dimeric. All right, so these are stronger because they are more extensive. These weaker interactions between the dimers allow them, okay, we are now talking about the polar bonds. Okay, the polar interactions, they allow the dimers to rotate relative to one another to form the different conformations of hemoglobin, the R conformation and the T conformation that we just talked about in this lecture, in this lecture slide. That the T conformation is the tense conformation, which is the low oxygen affinity state of the hemoglobin, in which the polypeptide chains are tightly held to one another and the bonding in between them is very strong. Whereas in the R conformation, some of the bonds break, and it is formed when one of the heme groups get oxygenated, and it interacts with the other heme groups to break some of the bonds, some of the polar bonds basically, and this allows more oxygen to this allows the oxygen molecule to bind more easily to the other heme groups. Okay, so that's why hemoglobin can get both, it can attain both the conformations, the R conformation and the T conformation. Okay, and this, as I told you, this changes in conformation is known as cooperative binding of the hemoglobin molecules, and it is possible due to the heme interactions of uh, adjacent polypeptide chains. Okay, heme interactions, and I told you that this interaction is not direct interaction between the heme groups because they are quite far apart from one another, but it is the indirect interaction with one another. It is the interaction of the globin chains. Okay, it is the interaction of the globin chains with one another, the surface interaction of the globin chain, which induces the conformational changes of the hemoglobin. Now let's move on to the next this slide. All right, this diagram is taken from your book Lippincott, as you guys can recognize it. All right, alpha beta dimer 1, alpha 1 beta 1 dimer, and alpha 2 beta 2 dimer. All right, so strong interactions, primarily hydrophobic interactions between alpha beta chains. They form stable alpha beta dimers. Okay, so we've just discussed that these interactions are pretty strong and they are hydrophobic interactions primarily, and they keep the dimer stable. And in between the two dimers, we have weaker bonds. We have ionic bonds and hydrogen bonds in between the dimers themselves. In between the whole dimers, there are relatively weaker bonds. Now, when the dimers get oxygenated, when any one of the dimer gets oxygenated first, what it will do is that it will induce a change in the adjacent dimer, and then it will also induce a change in the adjacent dimers, and it will try breaking. This change will break some of these weaker bonds, some of these ionic and hydrogen bonds between the dimers themselves. And this breakdown of the some of these bonds, what it will do is that it will cause these polypeptide chains to become a bit looser. Okay, the polypeptide chains will become a bit loose, and once they become a bit loose, so the conformation that the hemoglobin now attains is a more relaxed state. And after that one oxygen molecule has bound, then the subsequent oxygen molecules can bind to the subsequent heme groups more easily. Okay, so the first oxygen molecule that binds to this tight conformational hemoglobin, this deoxyhemoglobin, it is pretty difficult for that oxygen to bind to them. But once that binding is complete, then what happens is that the subsequent oxygen molecules, they can easily bind to the other heme groups as well, and in doing so, after the oxygens have bound, the oxygens are bound to the heme groups, the subsequent heme groups, then the polar bonds, some of the polar bonds break and allows the hemoglobin to attain a more relaxed conformation. Okay, this oxyhemoglobin is a more relaxed state of hemoglobin that has a much higher affinity for oxygen than the tense T state. Okay, so that's it for this lecture. And in the next lecture, we are going to talk in more detail about this cooperative behavior of hemoglobin and the R and T structures. Thank you.